DNA profiling methods are important in detection and can provide strong evidence for claims, for example, in paternity disputes. So instead of denying guilt in a song, Billie Jean is not my lover, she's just a girl who claims that I am the one, but the kid is not my son. If the kid is really not your son, you can support your claim with DNA evidence by comparing DNA variations present in your DNA and the child's. Since children inherit DNA from parents, variations seen in a child must come from mother or father. Besides relatedness, identity can be determined, as within a single individual, DNA is generally conserved throughout the individual's cells and in time. But DNA is most likely different between different individuals due to mutations, independent assortment and crossing over in meiosis, as well as the random fertilization of a particular sperm with a particular egg. Variations in DNA among individuals can be detected by sequencing the whole genome, which looks at every one of the billions of base pairs in each person's genome. Since an estimated 99.9% of the genome does not vary among the human population, it is easier to hone in on just the differences using DNA fingerprinting or profiling methods. A single variation, however, is not enough to differentiate individuals from each other. 13 to 20 tandem repeat loci with multiple alleles are often used in DNA profiling. The more loci are compared and the more alleles for each locus, the less chance there is that individuals will have the same alleles at all loci. Single nucleotide polymorphisms, SNPs, though having fewer alleles per locus than tandem repeats, are present more widely in the genome, so can also be used in DNA profiling. The early DNA fingerprinting method of Alex Jeffreys involved restriction fragment length polymorphisms, RFLPs, and probes for variable number of tandem repeats, VNTRs. Restriction fragment length polymorphism analysis begins with genomic DNA which has been extracted and purified, being treated with one or more restriction enzymes which cut the DNA whenever specific sequences of bases occur. In some individuals, random changes in the DNA will cause one or more restriction enzyme recognition sites to be lost or gained, so cause variation between individuals in fragment numbers and lengths. These fragments can be separated based on size by electrophoresis. For organisms with small genome sizes like the lambda bacteriophage, which has a 48.5 kilobase double-stranded DNA genome, restriction digest generates only a few DNA fragments. So if the colorless DNA on a slab gel is stained with a DNA-specific dye, distinct bands can be seen. However, for organisms which have big genome sizes, such as humans, whose genome size is approximately 3 billion base pairs, the digested genome consists of many fragments of the same and varying sizes. Thus, electrophoresis and staining of the digested DNA yields a continuous smear on the gel, which is not informative. So for organisms with big genome size, after electrophoresis, a labeled probe helps view the target fragments. A probe is a single-stranded DNA complementary to a specific region or regions of the genome. Using multiple probes simultaneously for different loci or multi-locus probes that hybridize to multiple locations leads to more informative results. Alex Jeffreys used probes to the variable number of tandem repeat loci, as VNTRs have many loci and many alleles per locus. VNTRs are 6 to 100 base pair units repeated up to thousands of times tandemly. Here are two examples of probe sequences for VNTR loci that have been used. GGA-GGT-GG-GCA-GGA-XG, in which X is A or G, or AGA-GGT-GGG-CA-GGT-GG. hybridization of probes to size fractionated DNA is more efficient if after electrophoresis, the DNA is transferred to a membrane in sudden blotting. The capillary transfer of fragments of DNA on the membrane means the fragments will be located as on the gel. The DNA on the membrane has to be denatured to enable the probes to bind to the complementary nucleotides on the DNA. After hybridization, the excess probe is removed, and the membrane exposed to X-ray film if radioactive labeled probes are used. Since the radioactive tags on the probes emit radiation that react with the film, bands will be seen on the X-ray film mirroring where probes are bound to DNA fragments. Interpreting results involves pattern matching of bands on the VNTR fingerprint to reference VNTR fingerprints. The pattern of bands is unique to individuals, as individuals have different restriction enzyme cutting sites as well as different combinations of the many mini-satellite alleles at the multiple mini-satellite loci. In identification, even if one allele at one locus is different, it can be concluded that the DNA is from separate individuals. Related individuals will have some bands in common, inheriting the same DNA variation from common ancestors. The bands on the VNTR fingerprint of a child must be from mother or father, as a child inherits half his DNA from each parent. Newer techniques for DNA profiling have developed. Short tandem repeat analysis is now widely practiced and uses PCR of what else but short tandem repeats, or STRs. PCR can amplify STRs more easily than VNTRs, as STRs are shorter, having a core unit of between 1 and 6 base pairs and repeats to about 50 to 300 base pairs. Also, STRs, like VNTRs, are present at many loci, each locus having multiple alleles, so providing sufficient variation for discrimination of individuals. The use of PCR means only a small sample of DNA is necessary. Even degraded DNA may provide results, and the PCR and microsatellite-based method results in data which are easier to interpret. Though the same repeat sequence may be present at different locus, each is amplified using different primers, as STRs are flanked by non-variable segments of DNA which differ between locus. The forward primer for each locus, if tagged differently, can help pinpoint alleles of different loci. To view the different sized fragments, electrophoresis is required to separate these fragments. Capillary gel electrophoresis enables small amounts of sample to be analyzed and fast analysis. When amplicons pass through a capillary gel electrophoresis system, the smaller fragments migrate faster and pass across the path of a laser beam first, followed by fragments of increasing size which migrate slower. The laser causes dyes attached to the amplicons to fluoresce and be detected by the camera, resulting in a series of peaks which can identify individuals and relationships. It must be noted that the size of peaks isn't related to the size of the STR allele. The height of the peak is just the intensity of the fluorescence, which might increase if, say, more fragments of a specific size labeled with the same dye are present. The size of the fragment is related to the time taken for the fragment to move the distance to the laser, and the size of the fragment may be determined by comparing to fragments of known sizes in the DNA ladder, which are subjected to electrophoresis at the same time. Identification and kinship analysis is similar to how it is done in the VNTR method of DNA fingerprinting, but if capillary electrophoresis is used, the alleles are represented not as bands a different distance from the wells, but as fluorescent peaks detected at different times. If all alleles of all locus seen in the two DNA samples are the same, the DNA source of both samples is probably the same individual, and alleles in a child must all be inherited from one or the other parent. Another method of DNA profiling is amplified fragment length polymorphism analysis. AFLP analysis does not necessitate knowing the organism's genome sequence to design specific primers and does not need probes with sequences complementary to the DNA of the organism being studied. AFLP analysis involves the following steps: digesting the extracted DNA with a pair of restriction enzymes, often MSC1 and EcoR1, ligating or joining the fragments to an MSC1 adapter and an EcoR1 adapter at each end by incubation of the digested genomic DNA with the two adapters and DNA ligase. The short double-stranded adapter sequences easily hybridize to the DNA fragments since each has an overhang that is complementary to the overhang on the DNA fragment generated by the restriction enzymes. Once the DNA fragments are sandwiched between the MSC1 and EcoR1 adapters, the fragments can be amplified using selective PCR primers complementary to the MSC1 or EcoR1 adapter sequences, but with up to 3 unique nucleotides added following the end of the original restriction enzyme recognition site. PCR with selective primers, that is, with added nucleotides at the 3' end, do not amplify every single genomic DNA fragment, which would be difficult to analyze, but a subset of fragments. One of the 2 PCR primers used is also radioactively or fluorescently labeled, which leads to the production of PCR amplicons labeled for detection. Depending on whether electrophoresis is on a slab gel or capillary gel, the DNA profile will be observed either as bands on a membrane or peaks in an electropherogram. So in summary, DNA profiling is used to identify an individual or relationships by comparing DNA variations such as VNTRs, STRs, or SNPs present in the different DNA samples. VNTR analysis involves DNA extraction, DNA digestion, electrophoresis, southern blot, probe hybridization, and autoradiography. STR analysis involves DNA extraction, STR loci amplification using PCR, electrophoresis of amplicons, and detection of alleles as bands from autoradiography or peaks in an electropherogram. AFLP analysis involves digesting genomic DNA to produce a population of restriction fragments, ligation of priming sites, selective amplification by PCR, electrophoresis of amplicons, and detection of fragments. DNA profiles or fingerprints detect relationships by looking for bands or peaks in common between individuals. For example, a child's profile will show bands or fluorescent peaks that match bands or fluorescent peaks in parents' profiles. DNA profiles that identify an individual to a DNA sample requires all the same bands or fluorescent peaks to be present in the DNA profiles. However, it must be noted that having the same alleles for a limited number of loci is not conclusive proof that the DNA is from the same person. By chance, 2 people could share the same alleles in, say, the 13 loci tested. Now let's review DNA profiling by answering some questions on the topic. Question 1. The different lanes of the gel shows the VNTR results of DNA samples taken from a crime scene and 3 suspects involved in the case. Which suspect's DNA was found at the crime scene? A. Suspect 1, B. Suspect 2, C. Suspect 3, D. None of the suspects. The answer is C. Suspect 3, as suspect 3's DNA bands are the same in number and distance from the well as the sample DNA obtained at the crime scene. Question 2. The electropherogram seen here shows the same 4 markers of the DNA profile for a man, woman, and child. The man and woman claim to be the parents of the child. A lawyer disputes their claim, saying that according to the information on the DNA profiles, this man and woman cannot be the parents of this child. By referring to the profiles, explain whether you agree with the lawyer. The answer to this question is that, based on these 4 markers, this man and woman could be the parents of the child. For locus D8S1179, the child could have received the 11 base pair repeat from the man and the 12 base pair repeat from the woman. For locus VWA, the child could have received the 18 base pair repeat from the woman and the 20 base pair repeat from the man. For locus D7S820, the child could have received the 8 base pair repeat from the man and the 10 base pair repeat from the woman. For locus D16S539, the child could have received 1 copy of the 12 base pair repeat from the woman and another copy of this same allele from the man. That is, all the child's alleles for these 4 markers can be inherited from this couple, so the child could belong to them. Question 3. You are given 2 samples of genomic DNA collected from 2 seemingly similar organisms whose genome sequences are not known. You are then asked to determine whether the 2 organisms are members of the same species. Which of the following methods would you use? A. RFLP analysis using probes. B. STR analysis. C. AFLP analysis. D. Any of the 3 methods could be used. The answer is C. AFLP analysis. Since in AFLP analysis, the genome sequence is not required to be known to design primers as in STR analysis, and probes complementary to the DNA studied need not be obtained as required in RFLP analysis. Question 4. Genetic differences in AFLP patterns between closely related species may be due to A. Mutation at a site recognized by one of the restriction enzymes used to digest the genomic DNA. B. Mutation of a selective nucleotide binding site. C. Insertion or deletion in DNA between adjacent cutting sites. D. All of the above. The answer is D. All the mutations described will change the AFLP pattern. When there is a mutation at a restriction enzyme recognition site, the restriction enzyme will not recognize the cut site, so there will be a change in the fragment size. When there is a mutation at the selective nucleotide binding site, the primer may not be able to bind to that particular fragment and amplify it. When there is an insertion or deletion between adjacent cutting sites, it will change the size of the fragment. Question 5. Here is an example of a microsatellite sequence that contains a repeat unit highlighted in red, a forward primer site highlighted in blue, and a reverse primer site highlighted in green. If you were to amplify the microsatellite by PCR and run the amplicons on an electrophoresis gel alongside a ladder to determine the amplicon size, how many base pairs long would the amplicon be? The answer is 131 base pairs, as the amplified region extends from the beginning of the forward primer to the end of the reverse primer. If you wanted to determine the number of repeats, you would have to know how many bases away from the repeat sequence the primer hybridization locations are and the number of nucleotides in each repeating unit. So that's the review of the topic of DNA profiling or DNA fingerprinting. If you found it useful, subscribe to the channel and watch videos on other topics on genetics. Thank you.